Hello student, good morning. Today we will start the structure of plasma membrane. Structure of plasma membrane. All living cells are separated from each other and from their external environment by an extremely delicate 75 angstrom unit to 100 angstrom unit equal to 0.00075 meter to 0.001 meter thick, covering milli thick, covering membrane invisible under light microscope. The covering is called cell membrane, plasma membrane, cytoplasmic membrane or plasma lemma. Gorter and Grendel scientists have described the 1925 populated that it was a bimolecule lipid layer formed mainly of polar lipids, phospholipids and sphingolipids with the double hydrophobic water heating tails of their molecules in both layers directed towards the mid-axis of the membrane. Their polar hydrophilic water-loving heads at the two surfaces of the membrane. Danelli and Dawson scientists have described in 1935 ascertained that the plasma membrane contained a good amount of globular proteins. In addition to the lipids, they postulated a limit protein bilayer model of membrane structure with hydrated globular protein molecules absorb upon both the exposed surface of the original bimolecule lipid layer. Later, in 1954, and Danelli further proposed that there were places where lipid bilayer is perforated by pores lined by protein molecules. Only polar solute molecules and ions can pass through these pores. Using electron microscopy, Robertson in 1959 observed that the plasma membrane has a characteristic trilaminar appearance in a dark light dark or railway track pattern. Each dark line consists of pleated sheet of protein on surface and sheet of polar heads on lipids molecules. The light central line consists of a bilayer of non-polar fatty acids. Chain of lipid molecules Robertson called the trilaminar structure. A unit membrane which is continuous with certain membranous structures present inside a cell. Modern scientists consider plasma membrane as a flexible dynamic fluid barrier whose component molecules interact with each other and with those of external and internal environments of the cell of required for the various functions of the membrane. This concept has been best explained by the fluid mosaic model of membrane structure postulated by Singer and Nicholson in 1972 and confirmed by Brecher in 1985. According to this postulate, the core of the membrane is lipid bilayer in fluid phase, serving as a relatively impermeable barrier to the passage of most water soluble molecules. Protein molecules of their complexes occurs in the membrane not in continuous layers but as separate particles asymmetrically distributed in a mosaic pattern. Many proteins called peripheral or extrinsic proteins are loosely bound at the polar surfaces of lipid layer. Other called integral or intrinsic proteins penetrate deeply into or even completely through the lipid layer. These proteins mediate most of the other functions of plasma membrane in addition to about 32% lipids includes some cholesterol and 42% proteins. The plasma membrane also contains about 6% carbohydrates and 20% water. The carbohydrates occurs only at the outer surface of the membrane. Their molecules are covalently linked to the polar heads of some lipid molecules, forming glycolipids, and to most of proteins exposed at outer surface, forming glycoproteins. Carbohydrates bound in this way to membrane components make up the so-called glycocalyx of the cell surface. Besides having pores, the plasma membrane usually bears numerous minute finger-like surface outgrowth called microvilli. Finger-like surface outgrowth called as microvilli, which increase the surface area of the cell manifold. Functionally, the plasma membrane is most multifunctional part of the cell. The, the plasma membrane is composed of two layers of protein molecules and two layers of lipid molecules. The lipids molecules occurs in chain in plasma membrane. Two males chain of lipids remain parallel to each other and term of bimolecular or double layered structure. This is the structure of plasma membrane. Okay, thank you.